Right, good morning. Welcome to Film 4. That's a good term, isn't it? So Film 4 is going to be basically around energy production as the first topic. And along with the production of energy, or followed by the production of energy, is the production of the gas, or getting the gas, the oxygen, into the furnace where the fuel is burned. And therefore low levels of oxygen, or hypoxia, is a critical subject to study. And en route to getting the oxygen in, as we transport the oxygen in the hemoglobin, very frequently the construction of the heme part of the hemoglobin is disjointed and leads to quite reactive intermediate substances called porphyrins. So we want to look at this because disturbances or high levels of porphyrins can cause a lot of bodily dysfunction and sensitivities to light of particular wavelengths. And we're going to spend a little time looking at some nutrients. And today's ones that I've chosen is vitamin A and vitamin D. So we'll see how we go during the day if we have time to do a little bit more in depth on any other ones. So as we stated in previous film excerpts here, life and health are dependent upon adequate nutritional intake. I think this applies more and more from the hierarchy of when problems occur, even mechanical problems, which tend to occur because muscles are weak, as we know in kinesiology, and muscles are therefore out of balance. If one muscle is weak and the other one is relatively hyper, this is when we have mechanical problems, dysfunction occurs. And the reason the muscle gets weak is often because there's a metabolic problem or systemic problem related to the meridian and possibly the nerve supply to that muscle. So frequently it could be an organ relationship or some imbalance in the system, and then the muscle associated with that organ becomes inhibited, and then the mechanical problem occurs. So at the end of the day, adequate nutritional intake is really paramount. Even in the case of when people are allergic, very often they're allergic because they're deficient in specific nutrients, and certainly when they're toxic, when they've got a toxic overload, is usually because they've got a deficiency of the nutrients that upregulate the detoxification systems, particularly the cytochrome P450s and the glutathione, sulfation, acetylation pathways, etc. So at the end of the day, the most important thing to maintaining life and health is adequate nutrition. And this is something we always think about with children being born or during pregnancy, having enough nutrition for the mother into the fetus and the growing child. And then we go through this period of time, from about 18 onwards into 35, 40, where it doesn't seem to really matter so much what we eat. We can get away with everything, late nights, partying, eating rubbish, and yet never really get ill or problems. But it appears around about 30, 35, things very slowly don't repair in the same way that they used to. So the replication of the DNA isn't as 100% as efficient in that transcription as it was when we were younger. By 40, maybe some symptoms are occurring and things are going twang a bit more often. And by 50, they certainly start to dry out. We get a little bit shorter. The gel of the discs begins to lose its jelly and become more hardened, and so we lose a little bit of height. By 60, we virtually got no nucleus pulposus left, so we've lost a bit of height, and we notice that when we jump, we get a jar in the head much more than what we did when we were younger, and we could leap off walls and things there. So as we get older, certain nutrients become more important, and most of those are going to be minerals, because we need to be able to replace the minerals that we're losing, particularly with bone loss. Sometimes it's going to be the vitamins, although very often, as we get older, we tend to eat better food. Nowadays we eat more salads and more green vegetables, 5 or 6 a day, etc. And yet somehow the health doesn't seem to improve that much unless you can really get those odd missing bits in. And that's where we can score to try and find out what the odd missing bit is, or the multiple missing bits are with our patients. So we divide the nutrients up into the basic food substances, and probably the premier food substance is protein, because at the end of the day, the proteins are what we're made of. but they also run all the enzymes in the body are proteins, and all the immune system is proteins. The immunoglobulins that run our immune system are proteins. And also, all our brain chemicals and neuropeptides are of protein origin. So how the brain talks to itself between one neuron and another depends upon neuropeptides or chains of amino acids. So we have amino acids, which are the basic building blocks, which we have 9 essential ones that we get from our diet. And then the other 11 we can, in theory, make from those 9. But sometimes with the metabolic pathway of making an essential amino acid into a non-essential isn't present sufficiently. And so we need to take the non-essential one as a supplement. So they become what's called conditionally essential. So amino acids build tissues, they transport molecules, things like albumin and globulin in the blood. They form antibodies, they form enzymes, they build chemical messengers, neuropeptides, which is a combination name of hormones and neurotransmitters. They are biologically important organic composed of an amine group at one end and a carboxyl group at the other end. The key component is carbon, hydrogen, oxygen, and nitrogen. So this is the structure of an amino acid, an amine group, an NH3 at one end, and a carboxyl, COOH at the other end. And then we have different structures to these, which make them into different chemical properties. And many of the amino acids are antagonistic to each other. So, for instance, lysine and arginine work in opposite ways. So if you administer one, you may find you displace the other one, so you have to muscle test. More recently, we found if you take tyrosine, you may displace tryptophan. So after a week or so of using an amino acid, you may have to bring the patient back and retest sometimes. And don't be surprised sometimes if the antagonist or opposite one is then showing a little bit deficient. So amino acids are not fixed as necessarily deficient all the time, the same one. So you may be deficient. I'm deficient in the one that shows up for me is lysine, which is a branched chain amino acid, but I have to watch it because if I take too much, then it displaces other amino acids. So I just take 2 a day and everything's fine there. But this is another incidence of when people get older, they tend not to burn fat so easily, and therefore you tend to burn amino acids when you're running out of glucose and glycogen in your energy production, and you start to move amino acids. And the biggest source of amino acids to burn is in the muscle tissue. And the major amino acids in muscle tissue are called branched chain amino acids. So as we get older, valine and leucine and isoleucine become very important, because if we become deficient in our amino acid pool in those, then we start to strip our muscles when we exercise. So this is the important thing. So having said that, we have different types. And you'll see here some are hydrophobic, which means they don't like water. They don't dissolve into water very well. And some are hydrophilic, in other words, they like it. Some are positively charged, and some are negatively charged. And the reason for this is that when we make proteins, when these amino acids come together, when there's a charged amino acid, it bends the shape of the amino acid. So the amino acid protein, or the string of amino acids, is not a long linear structure. It has bends in it, it has kinks in it. And the kinks in there are where there's positives and negative charges. So when something attaches to that certain point, it may change the polarity of the amino acid, of this protein, and then it changes its shape. And it literally can physically change its shape to the ligand or the substance that binds onto the protein. And this is important, of course, in cell membranes when we have receptor sites, which literally change their configuration when a substance bonds onto the receptor site and it changes the protein structure. And this mechanically then moves various tubules inside the cell, which then connect into the nucleus, which then opens up the gene. So the gene, the sleeve on the gene itself, on the chromosome, can be expressed, and it pulls the protein sleeve to one side to open up the gene itself to get it to work, which is very clever. So it's a mechanical matter of when we get genes to express themselves. So it's a matter of mechanics rather than just chemical effects. So these are the essential amino acids, and these are the non-essential amino acids. So if we remember here, the non-essential amino acids means the body can make it. We have one here which is of particular interest, which is called selenocysteine, which is an amino acid, but instead of having the sulfur in the cysteine, it's substituted in by selenium. Now, selenium substitutes by a particular gene, so this is no longer a sulfur cysteine, but a selenium cysteine. And this is one of the ways that the body uses selenium. We now know there's 25 different amino acid proteins which use selenium within their construction. So it's not just selenium cysteine by any means. The left brain, Dr. Goodhart taught us this some years ago, is that the left brain weakness, when people have difficulty with counting, particularly if they count, say, from 100 backwards in threes, and they go weak, which means they've got an underactive left brain, they tend to give the hydrophobic amino acids, and with right brain weakness, it's hydrophilic, and with the right brain weakness, it's hydrophobic, in other words, the ones that don't like water. So he was able to differentiate and, in fact, make up 2 different products for people with left brain, right brain, which is quite interesting. We find that generally, when we supplement amino acids, it's only one that shows up at a time. We don't tend to find we give multiple amino acid products because of this antagonistic rule that if you give a high dose of one, you may displace the other. So we find we give the one which is deficient, run it for a couple of weeks, maybe a month, and then retest and see what shows up at the end of the next time. So if you are relatively deficient, because very few people are deficient in amino acids, it's a matter of the balance of getting all 21 in the right proportions. And that if you've got a relative imbalance there, you just supply the one that is the deficiency and not just give the whole lot. Otherwise, if you give an amino acid complex, you can maintain the very deficiency that you've got or the imbalance. Okay, so the second most important food compound are oils that get into our system. And we need the fats to form all our cell membranes. These are all made of saturated and unsaturated oils in the cell membranes, which we'll talk about, especially in relationship to hypoxia. They're sources of energy, so they're very important sources of energy because we get more than double the amount of calories of heat from fats as opposed to glucose and proteins. They're stores of energy, so we can store them in our body fat. They act as cushions to protect the organs, and they act as insulators, and they basically keep us warm, which is very important. Because as we'll see when we look at energy, most of our energy goes into basically keeping us warm. And if we didn't have that subcutaneous fat, we would be losing heat to the environment very, very quickly, like a house with no insulation on it. And cholesterol and other oils are very necessary to build certain hormones, particularly the steroid hormones. So fats are divided into saturated fats and unsaturated fats. A saturated fat means there's no double bond in the chain, so we have a continuous chain of carbons with hydrogens attached. So these are fully saturated. Carbon has a valency of 4, which means that every bit has a connection. And unsaturated means there's a double bond here, so that means that the carbon to here has a double bond, so it's not fully saturated with hydrogen. You can hydrogenate this by putting bubbling hydrogen through, which is called a hydrogenated fat, which then turns this into the quality of this. So you make an unsaturated, stiffer and thicker, and form like the chemical properties of a saturated fat when it's hydrogenated. And this is what they do when they make margarine. And this has been shown now to be one of the biggest causes of cardiovascular problems of the 20th century. It was between 1950 when they introduced margarines after the war. They now accept the fact that hydrogenating oils was one of the worst things they could do for clogging up our arteries. So that's got a single double bond. If it's a single double bond, it's called a monounsaturated, and if it's got lots of double bonds, it's called a polyunsaturated. Carbohydrates form our major source of fuel, so they're the source that we burn as energy. They link with amino acids, so they form glycoproteins, and they link with fatty acids to form glycolipids. So these are complexes which are particularly work in the brain. We divide them into single sugars and disaccharides. single saccharides like glucose, disaccharides like sucrose, and of course longer starch is how we normally store it, or what we call glycogen in the body, in the liver and the muscle tissue, which are molecules of glucose joined together, which can easily be split, and then we can reform our glucose in when our blood sugar level drops. So we store our glucose as chains of starch in our muscle tissue and in our liver. And then we got the more modern findings of the last 100 years. We realize there's certain components within our food which are vital, and we're still finding these. And I think there's probably, even now, there's more vitamins to discover. I don't think we found all the vitamins that the human body needs. I don't know whether any of you watched the QI program. I was watching it the other day, and they were saying that in the space of 1 year, 10% of facts are proved to be untrue. In the space of 10 years, 90% of facts today will be proved untrue. So where it's fixed that there are only vitamin A, B, and so on, we're discovering now in a year's time and in 10 years' time, there might be vitamins of all sorts of natures or other findings. But it's really interesting because these are supposedly written in stone and science, and yet in 10 years' time, they'll probably be dismissed as almost myth. So vitamins, they are activated to form coenzymes. So these vitamins are coenzyme precursors, especially of the vitamin B group. They act as antioxidants, ones like vitamin A, vitamin C, etc. They're involved with blood clotting, like vitamin K and vitamin P. And they form part of cell membranes, like inositol and choline. When they're activated, and most of the 20 coenzymes, 16 of these are vitamin Bs. So we've got thiamine pyrophosphate, thiamine triphosphate, flavin mononucleotide, which is the B2, flavin adenine dinucleotide, which are the two active forms of vitamin B2, NAD, nicotinamide adenine dinucleotide, and the phosphate form. These are the coenzymes of B3, CoA, which is the coenzyme of B5, pyridoxal 5-phosphate, which is the coenzyme of B6, tetrahydrofolate, methylene, methynyl tetrahydrofolate or folinic acid, methylene, methyl tetrahydrofolate, and H4-biopterin. These are all active forms of folic acid, coenzymes for folic acid. Adenosyl and methylcobalamin are the two coenzymes of B12, and biotin, which is one of the vitamin Bs that doesn't have a number, is a active coenzyme we used in the urea cycle to metabolize ammonia into urea. Vitamin C is a coenzyme because it becomes oxidized and reduced and recycles itself. Alpha-lipoic acid, which is the almost universal antioxidant, which is fat-soluble and water-soluble. So we see this in cell membranes and in the cytoplasm. S-adenosyl methionine, which is the active form of methionine and the universal methyl donor. And CoQ10, which of course we'll be looking at when we look at energy production. So this is a laminate which should be available. We try and do most of our basic seminars when we produce a laminate for them, and these should be available over there. And this is the basic laminate on the active coenzymes. So when I have a patient who comes in who strengthens to pyridoxal 5-phosphate, I like to go back to the starter substance, the substrate, which would be pyridoxine. So my question really is, if a patient strengthens to pyridoxal 5-phosphate, are they B6 deficient, or have they got a problem of activating pyridoxine through? So if a person doesn't strengthen or muscle test to pyridoxine, but they do to pyridoxal 5-phosphate, then I know there's probably a blockage here, an inhibition in the conversion. And the major reason for this is a single nucleotide polymorphism, or SNP, or mutation in the gene, to activate the enzymes which do this. So one of the approaches we could do is to use a very, very large dose of pyridoxine with the hope we force more of it through in the process. This was the orthomolecular approach of Linus Pauling 50 years ago, to increase the concentration and amount of the substrate to force this through. What we would do now in functional biochemistry is we would say, well, the person strengthens to pyridoxal 5-phosphate. Let's look at the cofactors here, the enzymes, like magnesium and zinc and B2, and see if we can get the body to activate it itself. So, in fact, when we muscle test, we find the person strengthens to pyridoxal 5-phosphate, but weakens to pyridoxine. So, because this is in relatively high, and because they're not able to convert. Now, we know that these enzymes, these kinase enzymes here, are expressions of the gene. So the gene itself encodes for genetic material to make enzymes. Obviously, the gene encodes for a lot of other proteins, but all enzymes are proteins, and therefore are all responses of the gene. Now, any equation doesn't necessarily need an enzyme. We will convert naturally pyridoxine into pyridoxal 5-phosphate, but in much smaller amounts if the enzyme isn't functioning. So if we were to give the body pyridoxine and magnesium and zinc and B2, we probably would get some conversion. And of course, everybody will get some conversion to pyridoxal 5-phosphate even when they're deficient. Otherwise, they die pretty quickly because we wouldn't have any of the functions occurring of pyridoxal 5-phosphate deficiency. So we will make some, but some people do inherit the short straw from their parents of not being able to convert, let's say, pyridoxine into pyridoxal 5-phosphate. So they may either need to take the pyridoxal 5-phosphate as a finished product, or may show that they need magnesium or zinc or B2. Now, interestingly here, we need B6 or pyridoxal 5-phosphate in several of the pathways of converting folic acid. So of making H4 tetrahydrofolate into methylene tetrahydrofolate, we need the B6 or pyridoxal 5-phosphate. So we're lucky in our work in functional biochemistry that we can chase back what a person needs. So here's an example where we've got 4 or 5 possible nutrients. So we've got a person who could have pyridoxal 5-phosphate deficiency. They could have pyridoxine deficiency. They could be deficient in magnesium or zinc or B2. Or they could end up with an activated form of folate deficiency. So when they come in, what their symptoms are could be any of those 5 deficiencies. You see, so we're very lucky because with the muscle testing, we can say which one the person needs. So if they respond to, say, pyridoxal 5-phosphate, but they don't respond and strengthen to magnesium, zinc, B2 or folate, we know that that's what the patient needs. Okay? So we can break into the chain and see where the problem is. Whereas basically a nutritionalist has to go with the symptoms that the person is experiencing, and that person's experience may be that they've got methylene or methyl tetrahydrofolate symptoms of a deficiency, but the cause is because they get pyridoxal 5-phosphate deficiency. So we would say we'll give them pyridoxal 5-phosphate because that will then activate this in a chain reaction, you see. So we're very lucky with the work that we do. When I look back into 30 years ago, before I did any AK at all, I used to teach dietetics and nutrition, and it was all from a book. It was all to do with the symptoms of what the person had. If you've got dry, scaly skin, we give you this. If you've got this, we give you that, and so on. It was a cookbook approach. Now, the great thing about applied kinesiology and functional biochemistry is we can tailor it to the individual rather than just the symptoms. So that's the other side of the laminate, where we show the other remaining coenzymes here and how they're activated. And during the course of these seminars, we will, of course, be looking at how we make each of these active coenzymes. And then, not necessarily finally, but at this stage, we have minerals. And we're all familiar with the big minerals like magnesium and zinc. These are the big boys, as I call them. And we know that they're the big boys because this is what you guys buy. Okay, this is what I prescribe for my patients, and this is what my friends and colleagues in Australia and Russia and America buy. So all over the world, it's magnesium and zinc on the mineral side. Many countries are deficient in other minerals locally, and therefore the land has difficulty sustaining the population of growing the crops on that particular soil. For instance, in Australia, they have a land mass which is big enough to take 300 million. It's not far short of the United States. But it can't survive a population more than 25 million because it's not fertile, and therefore it couldn't grow. And before the Brits went over there 200 years ago, there were just a few Aborigines relatively, and they scratched a living out of what was there. But when the Brits came, of course, they then farmed it and populated it. But there's a crunch point where you start finding that you can't sustain the population anymore because you can't grow enough. And that means then you've got to import food in. And one of the big things my friends over there tell me that they're deficient is selenium. Selenium is almost extinct. You can see it in the plants. They have a yellow leaf, a yellow shape around the leaf. So botanists can pick out mineral deficiencies in soil very, very well. And vets can pick out these things in animals, in cattle, when they're deficient in particular minerals. And they put a plate up on the fence, and then the cow licks it. When they're copper deficient, when they're zinc deficient, etc. The problem is that we don't do that. No medic says, your zinc deficiency, let's stick a zinc plate up on there, and you lick that every time you go past the kitchen. So we rely upon the zinc from our food. And what we found, again, which was a bit of a surprise through the years, is that not all zincs are zinc, if you like. They are zinc, but it depends upon what they're compounded with and how soluble that is, as to whether if it goes into solution it gets absorbed much better, and does it dissociate from the anion or the compound that it's with. So for instance, zinc chloride is a very water-soluble form of zinc, and when it's in solution the zinc goes one way, they call the anion, and they call the cation, and the anion or the chloride goes another way, so it dissociates. That's called splitting up, and that's ultimately how we absorb a nutrient, a mineral. It has to be ionized. We can't absorb zinc chloride. So you might buy zinc gluconate, zinc citrate, zinc picolinate. We do not absorb it as zinc picolinate. We absorb it as zinc and as picolinic acid, as the separate things. So the important thing with a mineral is how soluble is it? And how is the pH? To get a mineral to absorb, it needs to be just slightly acid, about 5 to 6 pH, because that would mimic the stomach acid. That's why we have stomach acid. We have stomach acid to prevent infections getting through our stomach, to activate pepsinogen into pepsin, and most important is to ionize minerals within our food. So once we've ionized our minerals, we can then dissociate the anions and the cations, and then we can absorb the minerals. So a dry stomach, as I call it, when it's low in stomach acid, does nothing to help people absorb minerals. So antacid medication may actually limit, even if you take the best quality zinc picolinate and things, because it may not be in an acid medium. So if you want a good mineral product, it should be as soluble as the compound will allow it. Okay, because not all compounds are 100% soluble. It should have a pH of between 5 and 6. Okay? Some of them taste disgusting, in which case, thank heaven, we do capsules. And some of them are more soluble and are quite pleasant, in which case we do liquids. So minerals supply the major elements and trace elements that may be lacking in the diet. They act as catalysts. We call these cofactors. So a mineral is a cofactor to activating an enzyme, like the enzyme I just described, the kinase enzyme, which is magnesium and zinc dependent. They regulate the permeability of cell membranes. They maintain water balance and osmotic pressure. These are our major big electrolytes, so things like potassium and sodium regulate that. And also the activity of the nervous system is regulated by them, like magnesium and calcium ratios. So they influence the contractility of muscles. This would be how much calcium goes into magnesium ratios, and regulate the response of nerves to stimuli. And this is very important in the heart, isn't it? To make sure the heart beats regularly and doesn't have any abnormal beats. So everything to do with minerals and the circulation and heart beating, nervous side, is all to do with sodium, potassium ratios and calcium and magnesium. So by using those 4 compounds, you can tell if a person's got an excess of one of them or a deficiency. So if they've got a deficiency, they strengthen to it, and if they've got an excess, a strong muscle goes weak. So life depends upon keeping a good balance, or we call this balance, of course, homeostasis, but it's an ionic balance which helps to maintain homeostasis. That means sufficient levels of the pluses and the minuses in there, the positively charged cations with the anions. So ionic balance depends upon adequate nutrient intake, and these all depend upon getting the right minerals in their ionized form at the end of the day. So remember, even if you swallow the best quality zinc, is it the right one for you, and is it soluble? And we found that, surprisingly, There's quite a few different forms of zinc that can strengthen a person. We need to know which is the best one. So one of the techniques that we'll show you is how you determine which is the optimal form when more than one tends to strengthen. But frequently, it's only one form of zinc. So if a person's tested with zinc picolinate and they don't strengthen, you tend to say, oh, well, they don't need zinc. But in fact, they strengthen to zinc citrate or zinc ascorbate or whatever it might be. So sometimes we have to keep several forms of the minerals because a person will respond to one type and not another. There is, unfortunately, no one universal nutrient complex which will strengthen 100% of people with zinc. We've got a pretty good one called Smart Zinc, but that's a combination of 3 different popular forms of zinc. But there is no one that you can say is 100% definitive that this person is deficient in zinc that you can always give. Life is not as easy as that. But by blending some and making them into complexes, sometimes we can get good products. So to optimize human performance, in other words, to get the very best out of us, we depends upon getting optimal adenosyl triphosphate or ATP production in generally the mitochondria. These are the powerhouses of the cell. So we need optimal nutrition. And optimal nutrition means getting the right levels of the vitamins, the minerals, the amino acids, etc. Optimal oxygen delivery. And this is critical because very few people get PO2 max after the age of about 35. This is why sports people tend to go down. They're at the peak of their career in their early 20s, and then they sort of fade a little bit. By 35, most of them are hung up their boots as such, or the track shoes. Doesn't mean they can't play football, they can't run, they can't play tennis, but the performance isn't so good because their PO2 max is not as good as what it used to be. So we need to look at how oxygen gets into the mitochondria where it's needed, and the transport from here all the way through to the cell mitochondria. We need to consider toxins, which we've looked at previous seminars, particularly toxic metals, because these inhibit enzymes. We need to look at chemicals, how to detoxify chemicals, again, because they can have a detrimental effect on enzyme function, and the effects of radiation, from ionizing radiation or non-ionizing radiation. The absence of infections, we know that if we're running an acute infection, we feel pretty tired. And the reason for this is because we often switch from one production of energy to another. So we switch from aerobic to anaerobic, which allows us to rest and helps us to recover. If we're running a low-grade chronic infection, particularly a viral infection, well, this we know has an inhibitory effect on our mitochondrial energy production. So we feel tired most of the time there because we flicked again into a resting situation. So we've got anaerobic rather than aerobic. And a positive emotional state. I don't think anybody can optimize their human performance when they've got a negative emotional state. Certainly this applies to optimizing it with sports. If you have a negative attitude towards your sport or your performance, you're not going to actually perform with a positive reaction. So we'll see that very much this week in the final week of Wimbledon. Those people who've got the positive attitudes are more likely to be the ones to win.